Hello everyone, today I'm going to do the lab report 6 video for you. In the lab, I'm going to introduce you is the measuring lactase enzymatic activity we did on week 4 of Bio 110 courses. First, I'm going to introduce you the background information. First, what is enzyme? How enzymes work? Enzymes recognize particular reactant molecules called substrate and convert them into products. In an enzymatic reaction, the enzyme recognizes the substrate by its shape and by the position of its hydrogen bonding sites and binds the substrate to itself at the active site. Once at the active site, the substrate rapidly undergoes the enzyme catalyzed reaction and converts substrates into products. And then how does lactase work? The enzyme we will study in this experiment is lactase, and the reaction it catalyzes is the hydrolysis of the disaccharide lactose into the monosaccharides galactose and glucose, shown in this figure. Humans require this enzyme for digestion of lactose found in milk and other dairy products. The medical condition known as lactose intolerance results when the body produces insufficient amount of lactase to digest the lactose found in these foods. Lactose intolerance can cause intestinal discomfort and symptoms such as cramps, bloating, and diarrhea. And what is our substrate ONPG? To measure the lactase enzymatic activity, either the substrate or the product of the reaction must absorb light in the spectrum. However, none of the molecules in the lactase reaction do. To solve this problem, we are going to use another molecule with beta-glycosidic linkage that mimics the shape of lactose and can be hydrolyzed by lactase. And this is ONPG. The left figure is the molecular formula of ONPG, and the right figure is how its color reaction works. And now we are going to do the lab experiment. It's shown by video. Let's see. First, extract the lactase by crushing the lactase tablet into powder. Then transfer them into the tube labeled as suspension. Add 10 milliliter 100 millimolar PBS buffer to extract the enzyme. Then vortex the tube for 1 minute. Here it is the lactase suspension. Pipette 1 milliliter of suspension into 2 tubes respectively and put them into centrifuge. Set it as 1000 RPM for 1 minute. Pipette 500 microliter supernatant solution from each tube to another clean little tube labeled as lactase extract. Here is the tube containing 1 milliliter supernatant solution, and the design experiment is shown in the table including 2 control groups and 4 test groups, which ended as 30, 60, 90, and 120 respectively. Each test groups, we use 3 tubes to do 3 times of repetitive experiment. Here's the steps they conduct these experiment groups. pipette 200 milliliter of the reaction content from each tube to the wells of 96 well plate. Here is the finished 96 well plate containing 2 control groups and 12 tubes of 4 test groups. The color changed yellowish gradually. Finally, put the 96 well plate into the microplate reader. Measure the absorbance at wavelength 450 nanometer. Here's the result from the example experiment. And the result provided by our lab manager is shown here. Due to the COVID-19, we remote students can't do the experiment in the school lab, so we use the result conducted by our lab manager for data analysis. We can see from this figure that these are wells of group control A, control B, tube 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10, 11, 12. And these are their absorbance read by the microplate reader. We then need to do some data adjustment to decrease the error by calculating average absorbance from each test group and subtracting the average of control group absorbance. The adjusted absorbance is here. Here is the standard curve I plotted from the previous data. We can see that as time gets longer, the absorbance gets higher so that it can indicate that reaction time is an essential influence of lactase activity. And this is the references I did for this lab report 6. And thank you for listening. Bye.